Okay, hello students. Today is March 21st, 2018, and this is my second video for your actual lecture, which is in under N135 Unit 8. And before I proceed with this lecture, I want to show you that I have added two slides here, and these slides are actually a study guide for Unit 7, so you can do at your own time, and we can discuss this in next class. So last time we were talking about genetics. We talked about this. He's the father of genetics, so we are not going to go deep into this. I just want to run quickly to this slide. Okay, so this is when we say karyotype. Karyotype is just how all the chromosomes are laid inside a cell, in a cell nuclei. So how they're all arranged. Now, if you see here, first 22 pairs are called with numbers. They are known by numbers. And the last one, X pair, has letter. So all the first 22, they are called autosomes. And the last one is called sex chromosomes. So any disorder affecting the first 22 will have that word first. The first word it would have is autosomes. Autosomal, okay? So autosomal from autosomes. Any disorder due to the gene which is on the last one, which is the X or Y, it will be known as X-linked disease or Y-linked disease. So that's very important for you to know how genetic disorders are, their terminology. So it's very important. I don't want to repeat what I said last time. How 2X's decide the female, 2X chromosomes decide the female, and X and Y chromosomes decides the male. And now, if you see how they are arranged, the pairs, the number of pairs you have, they actually are linked with some very common diseases. If you look at chromosome number 4, then chromosome number 4 is linked with Huntington disease. Chromosome number 7, it is linked with cystic fibrosis. Chromosome number 11, it is linked with sickle cell disease, which is very common. Actually, sickle cell is a race-based disease. It's very common in African Americans. And then you have 21. Number 21 chromosomes, it's linked with Down syndrome. And we also have many X-linked diseases, which is very common, is hemophilia. So that's very common X-linked disease. Y-linked disorders, they are not that common, and they don't have many genes, so not many disorders are heard about the Y-linked. Now, so far, you know that why any genetic disorder is called autosomal, because they are concerned with the first 20 chromosomes. And why they are called X-linked or Y-linked, because X-linked disease or Y-linked disease is concerned with the sex chromosomes. Now, disorder is also called autosomal dominant and autosomal recessive. Now, when we use the word dominant, it means that what allele or copy of a gene is more expressed in the offspring. So whatever is expressed more in the child. So, there can be one trait which a dad is carrying, and that can be more expressed in the child. Or there can be one trait which mom is carrying, that can be more expressed in the child. Recessive means that parents are not affecting their child directly. There might be carriers, and so it can be, the disease can be a generation before that. The disease can be in grandparents. Okay, so That's the basic difference why we call them autosomal dominant and autosomal recessive. Um, Now, if you go to, let's go to slide 22. These are all the terminologies. I'm not testing you on these terminologies, but you do need to know what they actually mean. Um, uh, mm, I think we have talked about this, that why family history is so much underutilized in preventive medicine. So, or anywhere you go, whenever there is a patient admission, we need to ask them about their family history. I go to my doctor, I told them so many things about my family history, so I know, so my doctor can assess what I might inherit even with my kids. So, and then same way, for our purpose, family history is important to determine reproductive risk, diabetes, hypertension, many heart disease, cancer, they all run in families. Okay, so now you see here, let's go to autosomal. Okay, now here you see a picture which is autosomal recessive inheritance, and then both parents are carriers, whatever they have the disease, they are the carriers of the disease. So it is colored in purple here, both parents, that they are carrying a trait of that disease, but they are not diseased, like they have not affected themselves. They're just the carriers of those genes. And if they have four kids, two kids have 50% chance of, no, two kids have 50% chance of being a carrier themselves, and the other two have 25%. The one has 25% chance of getting it, and the fourth one has 25% chance of not getting affected at all and being normal. So this is just a percentage of, like, if the parents are carriers of these diseases, how it would get reflected in their offsprings. So if they have four of them, some kids would be carriers, some kids would get affected, and one of them, like, can be not affected at all. So that's how the autosomal recessive means. Um, and uh, it says is they can have a negative family history, like before mom and dad, somebody is actually carrying those traits. So, and then two copies of mutation needed for expression. That means they need to, both mom and dad need to have those carrier genes, so the child can be affected. It does not discriminate between sexes, and both male and female can have it. And it can also have between, in some cultures, first cousins or second cousins, they marry. So in that also, it can get reflected. So it's not about um, it's not about just in the family, but it can spread, like, horizontally too, like, not from, like, usually the family genogram is like from top to bottom, grandparents, then parents, then child. It can go sideways too, towards your cousins, from first cousin or second cousin, something like that too. You don't need to know that much in detail. I just want you to know what is the difference between the autosomal dominant and autosomal recessive. And we would be actually talking more about the autosomal recessive disorders. So sickle cell disease, you might have done in some of the lectures, or you might do in future lectures. Cystic fibrosis, which is like, the lungs have a lot of mucus in them. And if you go to the pediatrics floor, you will see some kids with cystic fibrosis there. Then I don't want you to be concerned with Tay Sachs and hemochromatosis. We're not going to do much. The most common, which NCLEX also tests you on, is PKU. And we'll talk about PKU in uh, through a YouTube video. So I'm gave that in the next slide. You have a PKU video, YouTube video. Uh, you need to uh, watch that video so you can take an idea. And when mom has these syndrome, when mom has a syndrome, maternal PKU syndrome, she has to follow a very strict diet so her baby is not getting affected by this PKU. It's the baby, they inherit an error in body metabolism. In our body, we have an enzyme called phenylalanine. If you magnify this picture, which is on this slide with baby and extra, it will tell you the exact spelling of phenylalanine, and then it is this enzyme is responsible for breaking down protein in our body. So if there is a deficiency, we can have a toxic buildup of this enzyme in body, and this enzyme, if there is a toxic buildup, it can be very brain threatening and can affect intellectual capabilities of the child. And it can also lead to other neurological problems with the baby. So the diet for baby or mom, if mom is affected, then diet should be actually, as it says in this, the small um, diagram, small picture I have posted here, is no meat, dairy, dry beans, nuts, and eggs. In fact, they also have formulas which have no phenylalanine, not this enzyme, phenylalanine free formulas for babies too. NCLEX is very fond of PKU, so I mean, I would ask you to go and watch this YouTube video, and you should know this picture, very well. And babies are, but in the newborn screening, when babies are born, within the first 24 hours, they are tested, to see if they have this problem or not. Okay. So autosomal dominant inheritance, one parent is affected, one parent has a disease. Okay, one parent has this disease, so like Huntington disease. So one parent has to have this disease in order to give it to the children. The most common we find is there is some kind of gene mutation in family. The women in the family is just called, those genes are called BRCA1 and BRCA2. Here, they are called BRCA1 and BRCA2. So if your mom has it, your mom's mom has it, that means it is autosomal dominant. It is being carried over from all the females in the family. I don't know if Angelina Jolie, she got her mastectomy done maybe because she had these genes. I'm not sure about that. So it's like one of the parent has to have disease if to be considered autosomal dominant condition. This is just example, for the other autosomal dominant conditions. I'm not asking you to memorize it so much. Again, the picture came, so get familiar with this picture. This is repetition of what I have told you already. So what I have told you already, I'm not going to tell you again and again. Let me tell you. Wait, there is a very good slide I added. This is okay. Okay, threats to embryonic and fetal development. Now, in this slide, what I want you to keep in your mind is that how genes how, if at proper time prenatal testing and postnatal testing is not done, then a clinician or the doctor or the OB or whatever it is, they will not be able to find the birth defect which a child might have if they are born on time. And so like the and also, if testing is done, there are many problems which were found can be kind of corrected within that time limit, or parents can be more prepared to know that what kind of child they might be having, and if they have a chance of consulting, and having some like having a abortion before that, they can do that. But that's all ethical things. But from here I want to know that chromosomal abnormalities can be known through prenatal testing. Another word in this slide is teratogens. Now, teratogens are any agent, any factor that can cause birth defect in the child. And the most common teratogens, which is, very much, next slide we can see. Most common teratogens is alcohol. So alcohol is something which is advised again and again. So the women don't drink alcohol at all during the pregnancy. There is no leeway time that I can drink earlier, later. No, you cannot simply drink alcohol at all. No smoking, no alcohol. And it's said that around 10,000 infants are born each year with many mental, physical problems because their mom had drinking problem. And it says in our country it's every 1 in 750 births, you have this problem. When you'll all go for your for your maternity rotations, in some hospitals they have full floor dedicated to those kids born out of the women who were on drugs, alcohol, substance abuse. And you can see those kids, and usually their facial characteristics is very kind of very significant. You'll be able to know. They have Down syndrome kind of appearance. They have small head circumference. They have upturned nose, and then they have low set ears. They have flattened features. And I remember when you all were learning Jarvis, we were talking about how the Down syndrome child looks like. Then also uh, also other factors. This slide which we are actually looking right now, it tells you exactly how in the CNS, all the colored areas in this slide, they are telling you that that is the most sensitive period of baby's growth of those organs. And if any alcohol, any teratogens is ingested at that time, it can have a big defect on that part of the baby's body, like third big CNS actually is throughout growing, but it's the most dangerous part in the beginning, third, fourth, fifth, as it's seen in here in the diagram. So it's very important for you to just take a look at this diagram, and see how as the pregnancy progresses, what organs are developing and what danger, a child can have if mom is not careful of what she's putting inside her body. Okay, so let's go back. Just wanted to show you that thing. Okay. Okay. Now prenatal testing. Now genetic testing. Genetic testing is very important, and in this genetic testing, the it's just like analysis of mom's DNA or chromosomes, and the testing can be done to find a diagnosis, like if there is any problem with the mom, or it can be also done if they want to do any treatment. If there's a problem, they want to do the treatment, or there is any other decisions to be made about the pregnancy. And and usually it's performed to uh, the doctor will tell, what if the fetus is born, that he can have this potential problem in his life, or mom can have this potential problem as she progresses in her pregnancy. And in the beginning stages, the testing is just a simple blood sample. It is just a simple blood sample, and it's called triple marker screening. So triple marker screening, and usually done between 15th and 18th week. I think your book says the ending week as maybe 18 or 22nd. Look at your book, what it says about the triple marker screening. But I do know it has to be between, in the beginning, like 15 week or so. 15, yeah. And I think I added this. Yeah. So when they have a blood triple marker test, they are usually, looking at three things. So three hormones, and then it is the three hormones is AFP, HCG, and estriol. Now AFP, alpha fetal protein, this is made by fetal yolk sac and fetal liver. Now, and during second and third week of gestation, this AFP is responsible for you can, during second and third week of gestation, if you test the blood, and if the AFP is elevated, it's high level of AFP, it might suggest that the baby has neural tube defect. We'll talk about neural tube defect a lot when we were talking about the fetal period, when the fetus is growing inside the baby. We'll talk in detail about that. But just so you know, if there is high level of AFP, it means that the baby can have a neural tube defect. It is like a structure before the baby's actual spine is being made. So, and they can have something called spina bifida, or they can have anencephaly. So AFP elevated has to do with neural tube defect. Another hormone they check with the simple blood test is HCG, and HCG is a simple glycoprotein, and it is produced by the placenta, and it is high during the time of implantation, and it drops around 8 week of gestation, and then again it drops again at 12 weeks. Now, if you have high level of HCG, then it can indicate Down syndrome. Baby can have Down syndrome. Then the third one is estriol, and placenta produces estriol too, unconjugated estriol. And if it is low, then baby can have Down syndrome. Okay, so I just wanted to give a little bit of overview about that. And with postnatal testing, PKU is done. Newborn screening, PKU is done, to assess whether the child inherited any, PKU from the mom or not. Okay, so that. And then if this blood sample has been done and there are problems, some problems are found, then the doctor will say, how about we do more testing? And then they do something called chorionic villi sampling, which is CVS. There was a very good card in my office which Professor Jarvis gave, and it has a gestational wheel on it, and it shows you at every week what kind of testing is being done. So the basic is that blood sample test, and if there is a defect found, then they would do a chorionic villi sampling, which is called CVS. C for cat, V for Victor, S for sugar. So CVS, and that can also detect the, Down syndrome, cystic fibrosis, and all these tests can be done within 12 and 15 weeks. And then they also do, apart from chorionic villi sampling, they also do something called amniocentesis between 12th and 15th week. And the needle is used to extract the amniotic fluid from the uterus with ultrasound guidance. So they make sure if they are putting needle, inside the mom's womb, inside the mom's belly, when she's pregnant, that it doesn't hit the baby. So they just want a sample of that amniotic fluid, and that can also tell, a lot of abnormalities which baby can have. Now, I would like to stop here, because what I'm doing is I am going to make sections of videos. So right now we just did genetics, little bit of prenatal testing and postnatal testing. Oh, I forgot one thing, right? Okay, Nagel's rule. Now, I just wanted to throw this here because Nagel's rule and GT-PAL system will come very, very late in your Unit 8. And by that time, I don't know if your concentration will be a little, kind of saturated. So Nagel's rule is it's a method of estimating date of delivery of the baby. It's an approximate method. That's why it's called estimated. And in this, you just have to ask the woman, and we are assuming here 28 days of menstrual cycle. And NCLEX also assumes it's a 28 days of menstrual cycle. So please don't go deep in that, asking if somebody has 30-day cycle, 31-day cycle, please. So just take it as 28 days of cycle. So you have to ask when mom comes to your office, and you're the RN, you ask when was your last menstrual period. You subtract 3 months from this date. You add 7 days, and whatever you get will be the estimated date of delivery. So I just gave you an example, like first day of last menstrual period was June 10th, and then you subtract 3 months from June, and you add 7 days, which will become March 17 will be the expected due date of pregnancy. When we'll do the review of this section, I'll give more examples for you to practice on. But I just wanted to give you this one so you can start practicing more questions like these if you want. Another thing is GT-PAL system. Now, GT-PAL system is an acronym to understand women's obstetrical history. This simple GT-PAL system can become very complex if you want to go very, very deep in this. Okay, I'm not making it very difficult at your level, GT-PAL, but this is so GT-PAL. Every letter signifies women's pregnancy history. So gravida, G means gravida. This is the number of times a woman has conceived, including any current pregnancy. So if a mom comes in your clinic, like if a mom comes to the clinic and she has 2 kids along with her and she's pregnant, so her gravida would be 3. So because 2 you already see there, and 3 she's pregnant, so it will be 3. Okay, so gravida. Then T stands for term births. Now you'll have to memorize this GT-PAL because it has a range, from what to what week. And I would say make a flashcard. This will help you. This is the number of times a woman has carried a pregnancy to at least 37 weeks gestation and has delivered. It doesn't matter the baby survived or not. So it is just that her pregnancy went till at least 37 weeks and she delivered the fetus. Now, preterm births, this is the number of time a woman has delivered after 20 weeks of gestation, but before 37 weeks. Okay, so that is preterm birth. Abortion, this is the number of time a woman has lost pregnancy, whether it was elective or spontaneous, before 20 weeks of gestation. Okay. Living children, the term states living children, but what it really is referring to is live births. Okay. So Susie is 38 weeks pregnant. This is her second pregnancy. She delivered a healthy baby boy at 39 weeks with her first pregnancy. What is this woman's GT-PAL? So she is 2 times pregnant. So that's why we get G2. She delivered a healthy baby boy at 39 until with her first pregnancy. So T is 1 because we know one pregnancy went till 39 weeks, which was beyond 37 weeks. So she is term birth, so that's why it's T1. We will not include the second pregnancy because it's still going on and we don't know whether it has reached 37. She has not delivered yet. Okay, so yeah, she's 38 weeks pregnant, but she's not delivered yet, so we will not. So the T1, so T would be 1. So this is the number of times a woman has carried a pregnancy to at least 37 weeks and delivered. So that's why T would be 1. Okay, because we only know the first time she delivered a baby, and the second one she is 38 weeks pregnant, but she has not yet delivered. Preterm birth 0, abortion 0, and living children. She delivered a healthy baby boy, so we know for fact. So that is L1. I'll give you more samples once I'm done with all these sections of this Unit 8. I will give you a long study guide for Unit 8 too, and some questions to practice. I can do as much as I can, but I cannot make you all study, because you have to be yourself motivated to kind of, do good in maternity and newborn. Don't get overwhelmed by the content, because it's a very vast content. Look for something which is very important for the nurse to know, because our job is important as a health educator, as a health advocate and health promoter. What if this information is not given to the mom? What if this information is not caught on time, and what danger it can pose to the baby or to the mom? So that's very important for you to know. What I'm doing is that I am dividing these videos in certain sections. So the first video is genetics and genetic testing. The second video will be fertilization, and third will be human growth and development. If we are there on Friday, then I can finish the rest of it. Otherwise, genetics, fertilization, human growth and development, physical and mental changes during pregnancy, and prenatal assessment. These 5 videos at least, I will try to put as much as I can by tomorrow. Making these like a half an hour video, making it kind of is not that of a problem. The problem is when it converts to QuickTime and then I have to upload it to my YouTube. So I am stopping this section now. The second video will be on fertilization.